Bismillahirrahmanirrahim. I am Dr. Aisha Rashid, Assistant Professor, Department of Pharmacy, the Women University, Multan. I am here as an instructor of your subject microbiology. I have shared my self-introduction, timetable, reference books, and the course content at your LMS dashboard. You can check from there. The first topic of your course content is the introduction to microbiology. So we will start with the basics. First, we will see what is the microbiology. If you focus on the word micro means small, like small, bio means life, logi means studies. So basically, simply speaking, what this word microbiology shows, studies of small living things. So we can define microbiology as branch of biology that deals with the microorganisms that infects human, the disease that they cause, their diagnosis, prevention, and treatment. All this comes under the microbiology. The branch which will deal with the various aspects of biology, like it will deal with the study of microorganisms, number one. It will also deal with the study of diseases that were caused by those microorganisms. It will also deals with the diagnosis of the diseases, prevention, and their treatment. If we talk about the branches or sub branches of the microbiology, it can be classified as bacteriology, virology, mycology, parasitology, immunology, and serology. Bacteriology, the studies of the bacteria, is referred as bacteriology. Virology, the studies of viruses. Mycology, the study of fungus. Parasitology, the study of parasites. Immunology, the studies of immune system or immunity. Serology, the study of the serum, that is referred as serology. So this is about the basic introduction of the microbiology, the branch of the biology that deals with the microorganisms, diseases caused by them in any biological system, their causes, their treatment, their diagnosis. All this comes under the microbiology. If we see the effects of microorganisms on the human, microorganisms effects can be beneficial or it can be harmful. Both type of effects can be seen by the microorganism in humans or in any biological system. What are the beneficial effects? It can be used in the food industry for the production of various types of products, like for the production of bread, wine, cheese, yogurt, vinegar, and many others. It also have beneficial effect on industrial scale, like various types of enzymes, amino acids, vitamins, antibiotics, vaccines, vaccines. These production also involves the microbial flora. Moreover, pharmaceutical industry, sewage treatments, all these includes various kinds of use of various kinds of microorganisms. The third field which is beneficial is agriculture field. In agriculture field, lot of microorganisms are used for the enhancement of the production or for the treatment of various diseases or situation. So here are some food, industrial application, agricultural application. These are three major broad classification where the microorganisms are used as a beneficiary or as a friend. There may be the harmful effects of the microorganisms, like it causes the food spoilage. Lot of food is wasted because of microorganisms. The most important diseases, many diseases in every biological system, like many diseases in humans, in animals. So diseases, bacterial diseases, viral diseases, or fungal diseases. So the effects can be broadly classified into harmful effects and beneficial effects. Then microbiology includes The study of, as I have told you earlier, these are the various branches of the microbiology. Bacteria, the study of bacteria, bacteriology, the study of fungi, the study of protozoa, the most importantly, the study of viruses, and the study of algae. These are the branches or the microbiology which includes the study of all these microorganisms. So if we divide living organisms, firstly, living organisms are of various classes. Animalia, all the animals or the higher biological life comes under the animalia. Then there is the plantae, the kingdom plantae. Under microorganisms or microbiology, there are fungi, protista, and monera. In Monera, there comes the bacteria. Under Protista, there comes the protozoa, algae, and under fungi, there comes the yeast and the molds. This is basically the classification of living organisms, and of our concern, this oval shaped red circle denotes the classification of living organisms. Now, the basic structural and the functional unit of life. I think we all people are familiar. The basic structural and the functional unit of life is cell. There are two types of cells, prokaryotic cells and the eukaryotic cells. We will see here various features of prokaryotic cells and eukaryotic cells, like we will discuss the organisms which comes under prokaryotes and eukaryotes. We will discuss size, nucleus, cell wall, plasma, membrane, and ribosomes. If we talk about the prokaryotic cell, what is the typical example of prokaryote? Bacteria, the true example of prokaryotic cell. Eukaryotic cell, algae, fungi, protozoa, plants, and animals. Prokaryotic cell, what is the difference between prokaryote and eukaryote? There is difference of the size. Prokaryotes are very small in size, while the eukaryotic cells are somehow larger. Size of prokaryotes is 1 to 4 micrometers, while eukaryotic cell, it is greater than that of 5 micrometer. The nuclear membrane in case of prokaryotic cell is absent. Only nucleus is absent. That is why that you can say there is no clear cut nucleus is present. Only nuclear material is present. While in eukaryotic cells are also referred as true cells. These have clear-cut nucleus which is bounded by the nuclear membrane. So we can say nucleus is present in case of eukaryotic cell. Cell wall. Peptidoglycan. In the cell wall of the prokaryotic cell, peptidoglycan is present, while the peptidoglycan is absent in the eukaryotic cells. Plasma membrane. In the plasma membrane of prokaryotic cells, sterols are absent, cytoplasmic streaming is absent, while the sterols are present and cytoplasmic streaming is also present in eukaryotic cells. Membrane enclosed organelles are absent in case of prokaryotic cell as that of nucleus, while the membrane enclosed organelles are present in the eukaryotic cells. The ribosomes are of 70S, while the ribosome in the eukaryotic cell is of 80S. These were the basic features or differences between the prokaryotic cell and eukaryotic cell. It is believed or it is thought that prokaryotic cell is the initiation of the life. Here is the diagrammatic representation of prokaryotic cell versus eukaryotic cell. Here you can see a bacteria. This is basically simply an example of prokaryotic cell. While this is an animal cell, this is the example of eukaryotic cell. If we discuss this picture, firstly you can see nuclear material is just scattered. There is no clear-cut nucleus with the nuclear membrane. Clear? Prokaryotic cell, here is the cytoplasm. Here is the cytoplasm, nucleotide. These blue strands are nucleotides. These are the small projections are the pili which help them to move. The ribosome, the smaller green colored granules are ribosomes here, while the cytoplasmic membrane, here is this one, cytoplasmic membrane. Their green color, this is the cell wall, and the most outer, this brown, is the capsule. This is the flagella, which helps them to move. So this is the overall structure of a prokaryotic cell. While you can see very defined structure in eukaryotic cell. Here are the ribosomes within this. Here is the clear cut defined, this one whole, the red color ball, this is a clear cut nucleus. This is the nucleus. Then there we have said the clear cut membrane bodied organelles. These organelles include mitochondrion, these include cytoskeleton, this include Golgi apparatus, then rough endoplasmic reticulum. These are all the features or the present within a true cell, a eukaryotic cell. Now, what are the characteristics of major groups of microorganisms? We have discussed various groups like bacteria, viruses, protozoa, fungi, algae. So we will discuss the characteristics of major groups of the microorganisms. Firstly, we will see the bacteria. The first and most important characteristic of a bacteria is their size. The size of bacteria ranges from 0.2 to 1.5 or by 3 to 5 micron meter. The important characteristics of bacteria are, as we have already discussed, these are prokaryotic cell, means don't have clear-cut nucleus or membrane-bounded organelles. Bacteria are unicellular, single cell is present. Their simple internal structures, as we have discussed in the previous slide in the figure, they grow on the artificial laboratory media. They have the capability to grow on the artificial lab media. Reproduction is asexual. Mostly simple cell division is involved in the reproduction. One cell will be divided into two daughter cells and so on. Practical significance of bacteria. Some of the bacteria causes diseases. Some perform role in the natural cycling of the elements and increase the soil fertility. Manufacture of valuable compounds in the industry, as we have discussed in the previous slides. This is the importance. It causes diseases. The understanding of bacteria will result in the understanding of diseases, their treatment or the cure. The agricultural fertility, or it also involves the bacteria. Manufacture of various compounds in the industry, in pharmaceutical industry, in textile industry, they also includes the utilization of bacteria. So this is the significance or importance of bacteria. Here are some pictures. This is basically classification on the basis of structure. If you see single cell elongated shape, rod shape, this is referred as bacillus. If a single cell of round shape, spherical shape, it is referred as coccus. If the cell is in the form of spiral shape, this is about bacteria, spiral shape, this is referred as spirillum. Spirillum. If this spiral shape is too long, that is a corkscrew type, you can refer as spirochetes. These are referred as spirochete. If these are in the shape comma-like, this comma-like shape, then these are referred as vibro. Then if large number of spherical cells are combined in the form of chain, then these are referred as cocci. The cluster of these sphericals is referred as a cluster of cocci. This is referred, if two spherical shaped cells are combined, then it is referred as pair of cocci. Simply speaking, round shape is referred as cocci. Then the chain of this bacillus shape is referred as chain of bacilli. Clear? Here is the microscopic image of a coccus. You can see large number of spherical shape. Here is the microscopic image of vibro. Then this is the rod shaped bacteria. Different type of, this is the cocci bacteria, spirochetes. Their microscopic image is here. The second important class of microorganism which we will discuss is the viruses. First, we will see the size. The size of viruses is very much small than that of the bacteria. This is 0.015 to 0.2 micron meter. Important characteristics of viruses: do not grow. These do not grow on artificial media. As we have said in case of bacteria, that bacteria can be grown on the artificial media in the lab. They cannot be grown on the artificial media. These always require living cell within which they reproduce and they grow. Obligate parasites, that they are always dependent on the other. Electron microscopy requires to observe. Bacteria can be seen by the simple microscopes, while to study the viruses or to see the viruses, always electron microscopy is required. What is the practical importance? What is the practical significance of viruses? This causes diseases in humans, animals, and plants. These also infect the microorganisms as well. Here are some diagrammatic depictions of the viruses. Here you can see the RNA material, and on the surface these are the capsomer of capsid. This is also another structure of the virus. Inside there is the DNA material. The outer surface is capsomer, and the glycoproteins, these pin-shaped glycoproteins, are also attached. There is a more defined structure: nuclear material, RNA, capsid is also present, glycoproteins in the form of the pin, then the membranous envelope. This is the structure of virus. The tail fiber, tail sheet, head DNA. Here, the one by one, the structure of this tail fiber is elaborated by this figure: capsomers and RNA, and this one, that is the sheet shown here, and then head or material shown here. This is the original images of the viruses by the electron microscopy. Tobacco mosaic virus shown in this shape, adenoviruses, cluster of adenoviruses shown in this shape, influenza viruses shown in this shape, and bacteriophages. Here, the sizes are also mentioned. Bacteriophages are of 50 nanometers, 50 nanometer, 50 nanometers. So this is, I guess, resolution power. Now the third class after bacteria and fungi, bacteria and viruses, is fungi. The typical example includes the yeast. The size ranges from 5 to 10 micron meter. The important characteristics include that these are eukaryotic cells, like these are pure cells, having clear-cut membrane-bounded nucleus and all other features. These are also unicellular, can grow on artificial media in the laboratory. They reproduce asexually by the process of cell division. By budding, they are also able to produce sexually. What is the practical significance of fungi or yeast? Some causes diseases, some are used as the food supplement, and these are also manufacture of the alcoholic beverages. These are also used in the manufacture of alcoholic beverage, which is very big industry. Here are some pictures of fungi. You can see the birds are here in this picture. The process of budding, reproduction by the process of budding. Here is also the picture of fungi. Okay, after fungi there comes the fungi molds. We have discussed yeast. Now we will discuss the molds. The size of the molds ranges from 2 to 10 micron by several millimeter. The important characteristics: these are also eukaryotic cells. These are multicellular, while we have said that yeast were unicellular. Many distinctive structural features. These are cultivated on the artificial laboratory media. Reproduction is asexually as well as sexually. Then the practical significance. These are involved in the decomposition of many materials. On industrial scale, industrial production of many chemicals like antibiotics, then they can cause diseases as well. This is the importance or practical significance of fungi. Here are the pictures of some pictures of fungi you can see. Then the next class of microorganism we will discuss is protozoa. The size of protozoa ranges from 2 to 200 micron meter. Important characteristics include eukaryotic cell. These are eukaryotic. These are unicellular. Some cultivated on the laboratory media while some are on intracellular. Important features. Protozoa are eukaryotic, unicellular, some cultivated on the laboratory media while some are intracellular parasites. Reproduction is by both means, sexual and asexual means. Practical significance, some causes diseases while food for adequate, and these also provides the food for aquatic animals. The major source for the aquatic animal for the food is protozoa. So this is the practical significance. Because of some interruption, I will revise it again. The size of protozoa ranges from 2 to 200 micron meter. The important characteristics, these are eukaryotic in nature, means these have clear-cut nucleus, nuclear organelles, and membrane-bounded nuclear material. These are unicellular. These can be cultivated on the laboratory media and also grow on the parasite, on the host or the parasites. Reproduction is by both means, sexually and asexually. Their practical significance includes that these causes diseases like other the microorganisms. These are the most important role of this protozoa is that they are major source of food for aquatic animals. So this was, my dear students, about the introduction. I will summarize. First, we have seen what is the microbiology. Then we have discussed the classes or branches of microbiology, virology, serology, bacteriology, like this. And then we have seen, we have discussed some size or important feature and significance of various microorganisms like bacteria, fungi, mold, protozoa. I hope you have understand this and this initial lecture will be useful for you people. I have shared your reference books on your dashboard. You can consult those books. If you have any query, you are also welcome to ask on your WhatsApp group, or we will have on a virtual class on Big Blue or Zoom on your mentioned timetable. You are always welcome to ask any question or to give your suggestion to improve this lecture. Thank you very much, hoping that it will be beneficial for you. I will see you in next lecture. Jazakallah.